Hello students, welcome to the academia. Today I will discuss the protein metabolism. Before that, we go to the introductory part of protein metabolism. Proteins are the most abundantly distributed organic compounds. The organic compounds means the compounds which have carbon in their structure. In 70 kg man, the protein weight constitutes 12 kg. The skeleton and connective tissue contains half. The body protein and other half is intracellular. Now, the protein turnover. The total amount of protein in the body remains constant, is equal to protein degradation. This process is called protein turnover, which is 300 to 400 gram per day. The proteins are made up of amino acids. So we can say the amino acids are the building blocks of protein. They are released by dietary and tissue protein. Mixed with free amino acids of body constitutes 100 gram. Glutamate, glutamine 50%, essential amino acids 10%, and the remaining are non-essential amino acids. The amino acids which are required for our body and what we get through the diet are called essential amino acids. The proteins rich in proline, glutamate, serine and threonine are rapidly degraded and have short half-lives. Amino acids are released by dietary and tissue protein. There is no storage of amino acid like carbohydrate which stored as glycogen and fat stored as triglyceride. If we intake excess protein, that is excess amino acid, they are metabolized and then oxidized to provide energy or converted to glucose or fat, and the amino groups lost as urea, which is excreted through the urine. Next, we see that how the body amino acid pool is maintained. We can find out in this flow chart. The body proteins are the source of the amino acid pool, as well as dietary protein, synthesis of non-essential amino acid. And from this pool we get again body protein, glucose, glycogen, fatty acids, ketone bodies, urea, porphyrins, purines and pyrimidines, creatine, neurotransmitters, etc. So this is how the amino acid pool is maintained in the body. The proteins which we take with our food daily basis, they are digested in our stomach and intestine. Proteins are too large to be absorbed by the intestine and must be hydrolyzed to yield amino acids. Proteolytic enzymes are responsible for degrading proteins, and the degradation occur in stomach and small intestine. In the flow chart, we can see the process of digestion by the different enzymes of pancreas and intestine. So in this way, we get the digested protein to amino acids. Then the amino acid through the blood, they are carried and goes to the different body cells where the metabolism starts. Here the digestion of amino acid in stomach is shown. Stomach having an important enzyme and pepsin and HCl, which is only secreted from stomach from the parietal cells. So now we see that how the amino acids are used in the different parts of the body. The valine, alanine, glutamine, how they are used and present in different parts of our body. Now we move to the important mechanism, that is transamination. So in today's video, I will discuss the transamination process also. Moving to the transamination. It is defined as a process in which amino group is transferred from an amino acid to keto acid to form an corresponding amino acid that is itself forming keto acid without liberation of ammonia. Here the enzymes are aminotransferases. All aminotransferases require coenzyme pyridoxal phosphate, and it is very important for the MCQs that this pyridoxal phosphate is very important enzyme for transamination process. So this is one example how the transformation occur. All amino acids except lysine, threonine participate in transamination process. The reaction is cytoplasmic and takes place in the liver. Basically, transaminases are introduced by glucocorticoids which promotes gluconeogenesis. So, what is the clinical significance of this? So, increased level in plasma indicate damage to cells rich in the enzymes like SGPT increase in toxic hepatitis, viral hepatitis and liver cirrhosis, SGOT which may increase in myocardial infarction, pulmonary disorders, etc. The transamination is very useful for synthesis of non-essential amino acids. Major oxidative deamination is catalyzed by glutamate dehydrogenase. In our next video, we will go through the process of deamination, which is of two types, that is oxidative deamination and non-oxidative deamination. So, this is all about the introductory part of the protein metabolism. If you people have any query or